Good afternoon, class. Now let's talk about other two pathways of complement system. Before starting with alternate pathway, let me just give you a gist of what actually happens in classical pathway. So you can see on the screen, the classical pathway, it requires first thing is that antigen antibody complex, right? Only then the components of C1 is going to get activated, which further is going to clear C4 and C2. Once it gets activated, it is going to get splitted up into two segments, that is C4A and C4B. This is going to stay with the antigen, whereas in the case of C2, it is a C2b which is going to stay with the antigen. Now this whole thing will act, this whole complex will act as C3 convertase. So once the C3 convertase, the C3, it is going to get split up into C3A and C3b. C3b, along with the rest of the components, it is going to form a complex which is known as C4b to A3b. Now this complex will act as C5 convertase. Now C5 convertase is going to split again C5 into C5a and C5b. Okay, now this from here it is a terminal pathway which is occurring. As I mentioned, it is going to sit on the antigen with all other components and deeply penetrate into the phospholipids of the antigen of the pathogen, and it is going to make pores over there, and which will gradually cause the death of the antigen, right? Now let's move on to the alternative pathway. Besides the classical pathway, complement system can be initiated by another method called alternative pathway. Unlike classical pathway, the alternative pathway is initiated by cell wall constituents of both gram-positive and gram-negative bacteria as foreign particles. Microbial surfaces directly affect the serine protease C3, gradually cleaving off C3 into C3A and C3B. This conformational change extends its effect on another factor, factor B. In turn, BA removed from the active site, keeping capital B, small b, towards the C3b in the presence of magnesium forms, and C3b, capital B and B, and considered as C3 convertase of the alternative pathway. So what do you understand by this? So besides the classical pathway, the complement system do have other ways also of sorting up the mess, right? So it is the classical pathway where you can see, as I told you just now, it requires antigen antibody complex for the component of C1 to get activated. But whereas in alternate pathway, it does not require an antigen antibody complex, right? What it needs is a constituents of both gram-positive and gram-negative bacteria. Another thing which you should remember in alternate pathway is that for the initiation of alternate pathway, the activation requires the presence of preformed C3b or form of C3. So it requires a preform. That is what is mentioned here. It requires a preform C3, C3b, right? Now what happens once the C3b is present? There is a slow process. As let me remind you, as I told you in my previous video, that C4 and C3 contains an internal thiol ester bonds. Okay, I hope you remember it. So what happens, there is a process called C3 tick over, in which few molecules of C3 will undergo a spontaneous conformational change, which will allow water molecules from the external to gain access to the highly reactive thiol ester So what happens, it is going to hydrolyze the metastable bond and forms C3H2O. Okay, so what you understand is that the C3, it has got the thiol bond. Now what will happen spontaneously, it is going to undergo a spontaneous conformational change, because of which a water molecule which is present nearby, it will gain access into this thiol bond. Now, as a result of which, it is going to hydrolyze the metastable bond and it forms a compound C3H2O. Now, it is not necessary that it has to combine with water molecules always. Any suitable acceptor molecule uh, which is present nearby during the conformation changes. It can go and bind to that active it can go and bind to the activated surface instead of water. That is, it is not necessary, it always has to be water. Sometimes it can be any suitable acceptor. If that is the scenario, now the C3 is going to go and bind to that activated surface instead of water and forms a nucleus for the alternative pathway activation. So the thing which you have to remember in the alternate pathway is that it requires preformed C3 or C3H2O, and you should know how this C3 is actually it forms to C3H2O, a process called tick over. Okay. Now, what happens once the C3b, okay, it is present. Now, there is something else which is called factor B. This factor B is going to bind to C3b or C3H2O. Okay, once it binds, and there is another factor which is called as factor D. It is going to split the factor B into the two segments, that is the larger segment and the smaller segment. Obviously, the larger segment is going to stay with the antigen, right? Now, let's move on to, so hope you understood. Let's move on to the next slide. Binding of C3b exposes a site on factor B that again serves as the substrate for an enzymatically active serum protein called factor D, right? Now, actually, factor D cleaves C3b bond factor B and helps to form the complex C3b, capital B and small b, right? So initially you need I'm back with this. Right, yeah. C3 H2O, right? Now what will happen? The B factor is going to come and bind to it. Once the B factor comes and binds to it, okay, I'm very bad with this, so let me just erase it. Okay, I'll just explain. Now you know that it has got C3b complex. Okay, now what will happen? The factor B, once the preform C3b is present, now the B factor will come and bind to that. Okay, now what happens, the next factor, which is the factor D, right? Factor D, it is going to cleave C3b bond to factor B. That, there is something you have to remember here. Factor D is active only when, or it can cleave factor B only when it is bound to the C3b, right? If it is staying individually, it cannot go and cleave it. It has to go and bind with the C3b. Only then it can cleave the factor B. Now the action of C3b, C3b, capital B, A, small b, is very unstable. It becomes stabilized by the presence of another exclusive serum protein, that is propidin. So what are the things you have to remember here? You need a preformed C3, right? Next, factor B and factor D, and then you need propidin in the pathway. In this pathway helps to increase the convertase activity period. So it has a major role as it is going to help the convertase, that is the C3 and C5. Okay. Now formation of, now you know this is the formation we are talking about. C3b, capital B and small b accelerates the auto catalyze of more C3 component. Now this will act, okay, it is going to catalyze more C3 components which are present here, present, and it is going to form a complex that is C3b, capital B, small b, 3b. Now this will act as C5 convertase. Okay, C5 convertase. So remember what. So initially this is going to act as C3 convertase, right? This is going to act as C3 convertase. And now this, once the C3 components which are present, okay, they get splitted out. Now the component is, this complex is formed. Now this acts as a C5 convertase. Though the structural basis of C3 and C5 convertase vary in these two pathways of complement system, but their mode of action is alike. So all, it's very simple. The thing which you have to remember is, okay, it is alternate pathway. It does not require antigen antibody complex, but it do require antigen. That is the pathogen. That is the gram positive or gram negative. Now, in order to initiate it, it requires preform C3 and it requires factor B, factor D, and properdin, right? Now, what happens? The C3, which is there, it is going to spontaneously, it is going to change, okay, its conformation, and it is going to allow the water to enter and it forms C3H2O. That process is known as, what is it? It's known as TIS, C3 tick over. Now, once you have C3b here, now what will happen? The factor B is going to come and bind to this. Once it is bound to the C3b, it is going to, and there is another factor which is known as factor D. It is going to come and it is going to split the factor B into usual two segments. That is the factor capital, that is a larger segment and a smaller segment. Now, the formation of C3b, capital B, and small b will actually act as a convertase, C3 convertase, and it is going to cleave or catalyze C3 components and forms C3b, capital B, small b, and 3b. As now, this complex will act as C5 convertase. Okay, now let's move on to the next slide. The next part is the terminal pathway, which I have mentioned in the classical. So I'm not going to go further into this. So here what will happen? The complex which has been formed will now go and bind to the C5, C6, C7, C8, and C9 respectively, resulting in the membrane attack complex forming, which binds to the antigenic structure surface of microbes, right? Now it is going to make pores, it is going to, water is going to go, ions are going to get out, and whole, what will happen? It causes the death of the pathogen, right? Now let me just show you this. Now let's come back. So here, as I told you, this is the alternate pathway. Here you require antigen antibody complex, but here you require microbial surface. You need this preformed C3, right? It is going to split it up, and what will happen? The factor B is going to come and bind to it. Okay. Now factor B, once the factor B is going to come and bind to it, the factor D will go and cleave this B factor, BAN. Now this will be the, as I told you, this will be your C3 convertase. It is going to again cleave the C3 which is present. Okay, now it is going to form C3b, capital B, small b, 3b. This will act as a convertase. And from here, as I told you, this will be the same thing. It is going to bind, all the other components are going to get, they're going to go stay with them together and they're going to make the pore, and then finally they're going to kill the pathogen, right? In order to stabilize, as the converters I have mentioned, it is not in a stable form. In order to stabilize it, there is another important thing which you should remember, that is propedine, right? Now let's move on to, yeah, the last one, that is the lecithin pathway. The third pathway of the complement system is the lectin-mediated pathway. Now, lectin-mediated pathway is activated by the binding of mannose binding protein present in the blood plasma to mannose containing proteoglycans on the surface of the bacteria and yeast it forms. So it just means like it is quite similar to the classical pathway. Now this mannose binding lecithin binds to carbohydrate residues on the surface of the pathogen, and then it undergoes certain conformational changes similar to what happens in C1Q. So it requires carbohydrate residue, which should be present on the pathogen. Now, once what will happen if it goes, conformational changes will occur, and now there is something. Let's move on to the third point. Yeah, MBP and MASP, that is mannose binding protein or mannose associated serum protease, right? Now, this MASP one, there are two types in it. Mannose associated serum protease are two types. The type 1, it is going to become active and it is going to cleave C4 and C2 to form C4b2A, right? Whereas the MASP2, MASP2, appears to be able to cleave C3 directly. So this thing you should remember, that the pathogen should have a carbohydrate residue. Once it has, what will happen, MBL is going to go and bind to that residue, which bring about a conformational changes. Okay, now this mannose associated serum protease are of two type, type 1 and type 2. Once it gets conformational changes occurs, the type 1 is going to get activated. When type 1, it is going to get activated and then it is going to cleave C4 and C2 to form C4b2A complex, whereas MASP2, which is going to cleave C3 directly. Okay, now in the listing pathway, MASP acts on the substrate of C4 and C2 component protein, right? So the rest of the thing, again, C3 directly gets cleaved, then the forming the resultant complex is going to act as C5 convertase, and the rest, everything is similar. So if you compare all these three, you just have to know a classical pathway. It is the antigen antibody reaction which is going to activate it. Here it is the microbial surface in the alternate, and in the lectin mediated pathway, it is the carbohydrate residue which should be present. Now, okay, now let's just wind up. Okay, just wind up. Now we can see a classical antigen antibody binds to a specific antigen on pathogen surface, right? Now here antibody binds to a specific antigen, it's a lecithin pathway, right? So it needs a carbohydrate residue here. And here antibody binds to a specific antigen on pathogen surfaces. Okay, now I think I don't have it. After a certain stage, you can see all this thing from here, it is going to be the same. So it's just quite simple. You just have to know few points which differentiate between these two. And after the first two steps and the rest of the step, it is similar for all the pathway. Right? Thank you.